Hi everyone, here are your Chapter 3 objectives, and I'm putting it here at the very beginning of the chapter. You'll have several short videos, one for each section of Chapter 3, and that'll be the way it will be for the remainder of the course. So these objectives cover the entire chapter, and objectives 1 through 3 are going to cover Unit 1, or Part 1 of Chapter 3. That's going to be name the 5 major shapes of bacteria, be able to recognize them, and you'll find those in Table 3.1. Know the arrangement of cocci, and this can also cross over to other shapes of bacteria, whether it be bacillus. Primarily cocci and bacillus is where you're going to see these terms. You want to understand the purpose of flagella, fimbriae, pili, as well as be able to compare and contrast the length and number of each of these on an individual bacteria cell. What is the purpose of and differences between a capsule and a glycocalyx? Name or know the components of the cell wall and the differences between gram-positive and gram-negative cell walls. And some of this material here will possibly cross over into the lab. What component in the cell makes a cell acid-fast? And I don't remember if you guys do an acid-fast stain in the lab or not. Then lipopolysaccharide, what is it? What are the different names? What does it cause? Where is it found? This really is a key point within microbiology, and being able to understand just the foundational knowledge of some of these bacteria cells. Then at the very end, we'll describe bacterial DNA, and then describe the purpose and the characteristic of a bacterial endospore. And real briefly, we'll get into sections 5 and 6, which we won't really test a whole lot on. It's still good information to look over. There's a few slides from each of those sections, but it's not a point within your chapter objectives. Alright, so 3.1 looks at the form and function of bacteria and archaea. And really what we're going to focus on throughout this course is going to be the bacteria cells. But we'll talk a little bit about the archaea in section 3.5. So all bacteria cells possess a cell membrane, a cytoplasm, ribosomes. The ribosomes are there to make proteins or facilitate the process of making proteins, a cytoskeleton to give the bacteria shape and structure, and then one or a few chromosomes. All right, then most bacteria cells, but not all, have a cell wall, and they have some sort of surface coating called a glycocalyx. And the glycocalyx can range in thickness, and so that's one variation between bacteria cells. Alright, this figure is a little hard to see on your screen, but if you look in your textbook, and hopefully you have a blown up picture of that for your ebook. Alright, figure 3.1 shows you the structure of a bacteria cell, and it's really nice that it makes layers here and points the specific line to each layer, and so it gives you a good idea of what the cell can actually look like. So this very outer layer is the capsule, the tan coating, and this is a slightly older version of the figure that's in your textbook, but this is the very basic one. So the glycocalyx is a coating or a layer of molecules external to the cell wall, and what it does is it serves as a protective layer. It can be sticky, and so it helps to allow a bacteria cell to stick to a surface, and it can also have receptor functions. All right, the glycocalyx, if it's a fit tight, if it's very dense, then it's called a capsule. If it's thick and loose and diffuse, then it's going to be called a slime layer. And so on this picture, it calls it a capsule, All right, but it's also called the slime layer and the glycocalyx, as you see here. All right, those words really are somewhat interchangeable within microbiology, even though in some cases the word used could be more correct than another one. All right, so other ones, all right, in all bacteria you have the cell membrane, all right, a thin sheet of lipids, all right, this yellow, yellowish mustard color layer, all right, the thin layer of lipid molecules. It's also going to have proteins within it, and it holds the cell. It's the, if you would, the bag within the cell, or the bag that holds all of the cell material. Excuse me. You have the nucleoid region. All right, this region is where you have the DNA, all right, the bacterial chromosome. Then you have the ribosomes. These are all these little circles that are going to facilitate protein synthesis. All right, the actin, cytoskeleton, these boomerang-looking or shaped molecules on this figure helps to give that internal scaffolding or structure to the bacteria. Cytoplasm, water-based solution. All right, the blue stuff here. All right, here is a plasmid. The plasmid is going to be a short circular piece of DNA. It's not found in all bacteria. All right, so here up here, some bacteria cells. Anything here, it's going to be found in some. All right, the plasmid is a double-stranded DNA circle, and it contains extra genes. These extra genes are not necessarily important to overall functionality of the bacteria cell, but generally carry genetic material that allows the bacteria cell to be more pathogenic. All right, whether it be an antibiotic resistance gene, and so then if the bacteria cell gets a hold of that plasmid, it can then be antibiotic resistant to whatever that gene is. It can also have a gene that might code for fimbriae. The fimbriae are these short hair-like bristles. You can think about it like the hair on your arms in terms of length and number. They're quite numerous. Those will add to its ability to adhere to other cells and surfaces. And this is important because a bacteria cell, if it gets inside of you, say inside your urinary tract, right, if it gets in there and it's just randomly there, it's going to get flushed out with the urine flow. If, however, it has the fimbriae, this allows it to stick tight to the side of that urinary tract, whether it's the urethra, inside the bladder, and then it sticks, and it's really like being able to grab hold of something, and then that flow of urine doesn't necessarily wash it all out. Then that bacteria cell can stay there, it can replicate, and those numbers expand to cause the number of bacteria needed to actually give a person an infection. So go through all of these different boxes and be able to put them in the right spot. All right? Is the material found in all bacteria? Is it found in just some bacteria cells? All right. And then the prokaryotic shapes and arrangements. All right, most prokaryotes, all right, the bacteria cells, and archaea in this case, but prokaryotes in general exist as unicellular organisms. All right, they can sometimes work together and act as a group, which you might see in a colony, on a petri dish, or in biofilms. Prokaryotic cells are very small, so they are about 1 micrometer to 750 micrometers. So there is a big range of size of individual prokaryotic cells. And then cells of one species may actually vary in the shape and size. And if that's the case, it's called a pleomorphic or pleomorphism, right? Morphism is going to be the shape, pleo meaning many. So there's going to be a variety of cells or shapes and size of this bacteria, even though if you look at it genetically, it's going to be identical. All right, here is table 3.1 that shows you the major shapes of bacteria. Up top, you have the cocci, or coccus. Coccus is going to be the singular, or coccus is singular, cocci is plural. All right, but these are going to be the circles. Then you have the rod or bacillus, and either way to use this, both of those words are going to be used interchangeably, but it's a rod. And you can see the slight variation that you would have in those rods, all right, within this shape. You have vibrio, and vibrio are rods that are gently curved. All right, so here's the boomerang shape almost. Then you have spirillum, and it's difficult in my mind, I have difficulty with this, of keeping the spirillum and the spirochetes separate, right? They are different, even though they kind of look very similar. Right? You look at this guy compared to this guy here, very similar. Spirillum are going to be rigid, right? It's a very rigid helix, and it's twisted twice or more along the axis, and it's very similar to a corkscrew. All right, the spirochete, however, is more flexible, and it resembles a spring more so than a corkscrew. So, I'm not going to have you memorize which one is which, because in all honesty, if you're going to memorize it, I'm going to have to memorize it, and again, I just battle which one is which. All right, they are both spiral shaped. They have differences, and we'll talk about a few of these organisms. So there actually aren't that many spirillum and spirochetes that are very common in microbiology in terms of a lecture course like this, simply because they aren't as numerous, and therefore the number that cause disease in humans not nearly as high. All right, then looking at arrangement, and I believe that this will be the last slide for this section. The arrangement is different than the shape, right? You have the shape of the bacteria cell, but then how does that individual cell, whether it be a cocci or a bacillus, interact with its neighbors? All right, so as mentioned a few slides ago, most bacteria cells are going to be unicellular. They're going to be single cells. But sometimes they do cluster, or they work with another cell, and that can be diagnostic in trying to determine what organism is what. All right, so here you have an individual plane or an individual cell, and you see this in one plane. All right, Diplococcus is two cells, and they just simply attach to each other, very similar to this, and that's what you would see on a microscope slide, right? Pairs of cells. So that's Diplococcus. Now, Streptococcus is going to be chains of cocci. If it was Streptobacillus, then it would be chains of the rod cells. So these are all going to be in a single or a single plane. All right, the individual cells, Diplococcus, Streptococcus. Now, if we look at the group of cells in two planes. All right, here you have a tetrad. Tetrad is going to have 4. All right, then you go to the Sarcina, which is a group of 8 to 64 cells, and this is basically a square of cells. And then you have the clusters, irregular clusters, and this is what you find with Staphylococcus and Micrococcus. And you'll probably deal with both of those in the lab at some point this semester. So the other thing, again, just to reiterate, the arrangement of cells is different than the bacteria shapes. So make sure you keep that in mind, and if you see a test question or a quiz question, that you make sure you are reading the right word and you have that lined up appropriate with the correct thought before you answer the test question. And that is the end for section 3.1.